All right, I guess I'll start then. My name is Karin Halsey, also known as meecier on various social media sites. I'm here to talk about do-it-yourself neuroscience, EMGs, which stand for electromyography, EEGs, electroencephalography, and a couple of other options for the do-it-yourselfer. Why do we want to do our own neuroscience? I don't really have to preach to this crowd about the importance of being able to take things apart and see how they work. But in addition, commercial equipment is expensive, especially research or medical grade equipment for these measures can run easily into the tens of thousands of dollars. But in reality, you can do your own setups for very little money, down under $10 in some cases. And the rest you guys know. I'm going to go into a little bit of detail in terms of an anatomy and physiology review, not as in-depth as I did in my talk last year, but if I gloss over something too much and you don't understand something, please flag me down. Keep in mind that I have tunnel vision when I'm nervous, so you might need to wave your hand really a lot before I notice you. The first thing to keep in mind is that many animal cells, and in fact plant cells as well, are electrically active. These currents are produced by a controlled movement of positive and negatively charged ions across cell membranes and from cell to cell. I'm just going to do a tiny bit of history, but the study of electrophysiology and electricity have been intertwined from the start. Galvani noticed in his very early work in the late 1700s that animal tissue was both electrically conductive and worked as a detector of current because he could string a frog leg into a circuit and it would conduct through and complete the circuit, but it would also flex in the presence of current. And Volta, with his invention of the battery, was one of the first to look at the effects of DC current on animal tissue. He was also particularly fascinated with electrical eels. A little bit more of the background. An electrically active cell at rest maintains a negative potential with its surroundings. And in fact, actually, most cells, whether they're electrically active or not, do the same. And this is partially due to the presence of negatively charged proteins within the cell. But in addition to that, there are a number of ions that are maintained against gradient in various concentrations. They're maintained through ion pumps, ion channels, and it's a very carefully kept balance at rest. And the electrical event of a cell is called an action potential. There is an entire cascade of events that happen with ion channels opening and closing that makes the cell potential shoot up to something relatively positive very briefly. And then return to the resting potential and actually undershoot that resting potential a little bit. That undershoot has a couple of different functions. One is that it creates a refractory period, prevents the cell from firing again right away. But the other thing it does is it prevents the action potential from traveling the wrong way or backwards up a neuron. That'll make a little bit more sense in just a minute here. Those action potentials are usually described as I showed them, and it looks like a static event happens for the whole cell. But in fact, it actually does travel down the length of a neuron from the cell body to the axon. Another thing you should know about that action potential is that you often see that curve. It's a very idealized action potential. Action potentials from different tissues and from neuron to neuron look quite different from one another. They might have little blips at the start. They might undershoot more. They might undershoot less. Especially when it comes to cardiac cells, that length of time where they're maintained positive is longer, so it doesn't look like a bell curve quite so much. So there's a lot of variability. Another thing that you should know is that an action potential or a cell firing for any given cell will look the same every time. And that sounds sort of basic to start with, but it has some interesting implications. One of which, and if you look across these, the top line where it says action potentials, those are the same things that I showed you just now, except compressed in time, so they just look like a spike. You notice the amplitude of those never changes between the weak stimulus and the strong stimulus. What does change is the firing rate, and it increases with a stronger stimulus. You'll also hear me talk tonight some about motor units. A motor unit consists of a neuron plus the muscle fibers that it's attached to. They do work together as one electrical entity. So what happens when you go to contract a muscle is that the action potential travels down the neuron and to the muscle fibers. And then the current travels along the surface of the muscle fibers, enters down into the muscle cell through a structure called the T-tubules, and from there triggers the chain of events that cause the actin and myosin filaments to move against one another to cause the muscle contraction. I'm going to talk a little bit about the difference between near-field recordings and far-field recordings. Near-field recordings are not really practical for use for the do-it-yourself or on human subjects, mostly because people object to being stabbed with needles. But it is possible to do as a do-it-yourself, or you can work on insects, you can work on other subjects, you can take a look at plant cell potentials, all of that type of thing. There are resources for this for the do-it-yourselfer if you're like me and don't necessarily have the know-how to start from the very basics. There are kits that come from Backyard Brains called spikerboxes that are assembled in the Ann Arbor hackerspace, actually. And they've got a number of experiments on their page with suggestions of experiments to do on cockroaches and earthworms and things like that that most people would be reasonably comfortable working with. For near-field recordings, I'm going to go back one. You have to use a needle electrode. So any type of conducting needle works. The finer the better, because what you want to do is record from one cell or possibly just a couple of cells that are close by one another. Things that you can do on humans without them objecting too much involve surface electrodes, typically. So we're making recordings through the skin on some part of the body. And by necessity, that means that you end up recording from more neurons at once, or more motor units at once. So an EMG recorded with surface patches is actually a number of motor units that are firing together, so you get a fairly complicated signal. With a little know-how, you can break that down into individual units, or maybe not, depending on just how good your recording is or how good your signal processing is. A couple things to know about EMGs. The firing rates of motor units vary quite a bit from cell type to cell type, depending on what kind of muscle they're working with. Things like that, anywhere in the range of 7 to 20 hertz. The amplitudes range greatly as well, again, depending on the muscle types and things like that. Unlike most sensory neurons, there's very little spontaneous activity in motor units. So when a muscle is at rest, those nerves are not firing at all. Whereas most sensory cells, there's a certain amount of spontaneous activity, and it tends to have something to do with how sensitive those sensory cells or those neurons are. And like I showed you in the one diagram, the firing rate increases with load for any one motor unit. The heavier or the more you need to contract or the more force that needs to be applied, the faster they will fire. There is also a phenomenon called recruitment, in which more and more motor units start working together to contract a muscle. Another thing that's available out of the box to use is a Backyard Brains toy, basically called an EMG spikerbox. And it's relatively straightforward, but you can record potentials from muscles using their little box. The data analysis options for the Backyard Brains devices are actually quite varied and useful. They have output via a speaker that's built into the box, so you can actually listen to it and tell whether you've got a good electrical connection and electrical recording going. They have output from data cables to the audio inputs of a computer or a smartphone. You can do data capture either with a program called Audacity, which is designed for recording sounds, but it works quite well for this application as well. Backyard Brains themselves has an app that's good for both Android and iPhone. And it is basically turns your iPhone or your Android phone into an oscilloscope, which is pretty nifty and useful for a number of different applications. And then, of course, if you're a coder at all, capturing from the audio input is pretty easy. You don't even really need to invent the wheel for this one. There are a number of libraries out there for you to pull information from. Troubleshooting is fairly problematic when it comes to EMGs. The biggest problem that people will run into is poor connection at the point of the recording electrodes. Some things that can help with that are a conducting gel, which you can make yourself. I actually have the components for it with me today, so if people want to play with my toys afterwards, we can make some conducting gel and hopefully get something working fairly well. You can use better electrodes, and in medical applications, they tend to use very fancy electrodes with silver or gold or something to really create a good contact. It's not strictly necessary for the level of work that we want to do, but it does help for sure. Another thing in terms of electrical connection is where you place your ground electrode. It needs to be in a non-muscular area so that you're not getting interference from a different muscle firing. So they suggest like maybe a metallic bracelet that you clip to or a wedding ring, someplace that's far away from the muscle that you're recording from. Other interference sources or noise sources are definitely problematic. You can get AC current noise, you can just get general room noise, ground loops in the building around you. It's kind of scary, the kind of stuff that interferes with recordings. So things like Faraday cages, electrical isolation really help. If you are recording data through a laptop, you want to be running it on battery and not AC. And also, if your screen is not running at full brightness, that can cause problems too, because that can create some interference as well. I'm going to switch gears at this point and stop talking about muscle recording so much and start talking about EEGs. EEGs, if you are familiar with the hospital version of these, this is going to look fairly different because most of what we do for do-it-yourself have a very limited number of channels, either one or two, as opposed to the hundreds of channels that you see people in the classic look. But either way, you end up looking at a composite signal, much like the EMG. You are looking at basically all the neurons in between your recording electrodes, firing in combination or firing separately. So it's a complex signal. The most common analysis that people do with EEGs is the typical brainwave analysis. They break it down into the spectral region. So the high frequency noises are what people generally associate with alertness or concentration. The low frequency is relaxation or even sleep. And any combination of those may indicate different mental states. People have a lot of fun with stuff like this. It's a nice source for biofeedback if you're trying to change the way you're thinking about things, stuff like that. Another thing that you can do with EEGs is leave all the data in the time domain and do what's called an evoked potential. And what you can do is you can look at the brain's response to stimuli, whether that's auditory stimulus, visual stimulus, or tactile even. And so you present the stimulus many, many times, and you record the associated EEG. And as you average that EEG over those many stimulus repetitions, all of the activity that's not related to the stimulus starts to average down to 0. And what you're left with is a waveform that's associated with the processing of that stimulus. So the example that I have here happens to be a visual evoked potential, but you can get similar things with different stimuli. Things to keep in mind with this is that you do need a fairly decent sampling rate. This happens at relatively short latency. This is on the order of 200 milliseconds or so. So what's available for the do-it-yourselfer? If you want to be out of the box and running right away, there's a company called Neurosky that makes headsets for as little as about $100. that are single-channel EEG recordings. They include with their box links to and projects for things like control of a radio-controlled helicopter or control of an electric wheelchair, just with changing your mental state and changing that focus versus concentration activity that I showed in the beginning there. This is what their basic out-of-the-box app looks like. But there are other methods of analyzing their data. Unfortunately, everything I've found so far is through Neurosky and their interface, but they have a number of apps as well that can help you export that data in relative real time. So you can do a fair amount of analysis after the recording as well. If this isn't to your liking, there are a number of resources out there for building your own EEGs. There are people who are just obsessed with this idea, and it's pretty cool, actually. The biggest conglomerate of information is the Open EEG project. Left the link up there for people who are interested. There is far too much for me to summarize here in terms of information. But they range from very low cost, under $10, you know, build it from the ground up yourself, to people who actually have fairly sophisticated setups using very high fidelity auditory recording equipment that allows for very good sample rates and data capture. So if you put electrodes on the skin, how is it that you can tell if you're recording from a muscle or from a neuron? And the sad news, unfortunately, is you can't, unless you start from a point that you know and can make some educated guesses about what it is that you're seeing. So what do you do to limit competing signals? When you're working with EEGs especially, there are a number of muscular artifacts that need to be kept in mind. Probably the biggest is eye blinks cause a huge artifact that can look like, apparently, I'm told, some sort of seizure activity. So people can get really upset if you blink your eyes a lot during an EEG. But if you know what those eye blinks are and you can filter those out, you can definitely reduce the effect of or the problems that you get with your data. You also can get artifacts from heartbeats. As you are probably aware, the cardiac muscle is highly electrical active. And even doing things like changing your electrode configuration can help with this to some extent. But if you record a simultaneous EEG along with an electrocardiograph, you can use that to indicate where those heartbeats are happening, and you can filter those out of your data as well. Some other things that people run into, if you are doing those evoked potentials in the time domain, you will get a stimulus artifact depending on the source of your stimulus. It's especially noticeable if it's an electrical stimulus, but pretty much anything that can cause interference will cause you issues. And again, you can also get artifacts from the electrodes themselves and poor grounding. And one of the things that I want to talk about with muscle movement, a fun fact is that your tongue is differentially charged from its base to its tip. So moving your tongue around during an EEG can cause some really whacked things to happen as well. So options for improving your data, a careful electrode placement and ensuring a very good connection. Ask the subject to hold still. You can test subjects while they are sleeping. Now, obviously, this will affect their brainwave analysis. But if you're looking at evoked potentials, those will be there whether someone's awake or asleep, assuming that they can still perceive the stimulus. I mean, if you're using visual, you can have to prop an eye open or something, but you know. A very, very important concept with do-it-yourself EEGs is that you need to impedance match your electrodes with your amplifier. Because if you don't, you end up with a DC offset, and it doesn't take much of a DC offset before your data starts getting clipped and sometimes completely useless. So really, this can't be emphasized enough. This is what most people spend most of their troubleshooting time doing. A very useful thing to do is to amplify your signal. It really helps improve the signal-to-noise ratios and all of that good stuff. But again, you need to do this with quite a bit of caution because that electrical isolation of the patient and amplifier is highly important. So any feedback of electrical current through your patient would be just bad. I'm going to talk some about how medical and laboratory grade EEGs are cleaned up even further. Again, this is not recommended for do-it-yourselfers, but it's good information to have. For laboratory and medical use, they often will use sedation and or neuromuscular blockers. Some cautions with that is that all drugs will affect your data. They do have effects on neurotransmission, and it will change things. And how much it changes things really depends on what you're looking at, what neurons are involved, things like that. Neuromuscular blocking will cause paralysis and death since you can't breathe without voluntary muscle control. That's why it's not recommended at home. But just for your information on how it works, because it is fairly interesting, is that within that motor unit, in that junction between the axon and the muscle fibers, the neurotransmitter that's used to communicate between those two is acetylcholine. Neuromuscular blockers, usually, most of them, work right at that junction, preventing the acetylcholine from working properly. So the action potential travels down the axon like normal and fires, but the signal never makes it the jump across to those muscle fibers. I have a bunch of disclaimers because I brought stuff for people to play with. I am not a professional at any of this. I'm not even particularly knowledgeable. I Google very well. Please research, if you decide to play with this, please research any risks and pitfalls for yourself. But I have here a summary of the 3 demos that I have here. They are all commercially available. I have not hacked them from their intended configuration for the purposes of this demo because I'm a little chicken that way. I have full intentions of taking these apart and playing with them more, but not for other people to play with. I did make a spikerbox from Backyard Brains. Mine is actually the single-channel version. This is the 2-channel version. But it basically looks like this. They say that it takes about between 2 and 6 hours to put together. I am a newbie. I knew how to solder, and that was pretty much it, and it took me less than 2 hours. Their instructions are very good, very clear. It was very satisfying to turn it on and have it work the first time. That was pretty awesome. What I've done so far with it, I've built it, I've turned it on, I've ensured that it works. I have it here today. You can take a look at it. I did not bring suitable test subjects because I thought that my roommates would object to me bringing cockroaches or other bugs along with me. But this is something that I do want to play around with quite a bit. I also have the EMG spikerbox from Backyard Brains. It is ready to play with. Please be gentle with my electrodes if you play with them. It is fine if they all get used, but it'd be nice if a number of people got to use them. I have so far been able to record successfully from it. People can play around with these. What do I want this for? I want it because it's basically a differential amplifier. So you are not limited to EMG recordings with it. And it occurs to me that this would be a perfectly suitable EEG setup if I could get sufficient amplification through it. So that's the hack that I'm looking at. And I also have the entry-level headset from Neurosky. I didn't break out the big bucks for the beefier and more comfortable models. What I've done, I've used the intended apps to look at the data. I will be honest, I found them a little bit buggy and a little bit annoying. Trying to get the raw data to output has not been successful for me so far. I can get the spectral analysis over time to output just fine, though. What do I want to play around with these? This is the thing that screams at me about being taken apart and seeing how it works. I would probably try to get it away from Bluetooth and into some other data transfer method. I would like to figure out if it'd be possible to do evoked potentials with a headset like this once it's been modified. In its current iteration, it is not. It doesn't have a suitable sampling rate to output the data. And I'm going to leave a resources page up there for a bit. These are the companies that I bought it from and the Open EEG project. My Gmail address in case you wanted to contact me, or I am also on Google+ under the same email. And I'll go for questions now, and if not, for whatever time we have the room, we can play with the toys or we can move someplace else and play with the toys. question. Yes?
Given limited equipment and all that, can you talk a little bit about, so what are some potential cool things that one can do with it, like the EEGs specifically?
The EEG specifically, I thought that the Neurosky induced suggested experiments are something that would appeal to most people from the start. The radio-controlled helicopter that you could make move left or right based on thinking a certain way. I think biofeedback's fairly interesting. I would like to be able to use, the reason I like Evoked Potential is I work with them at work with medical grade equipment, and if this is something that I could replicate on a budget, that would be really kind of cool, and I'd really have a deeper understanding of why it works, which is sort of my motivation. Evoked Potential is the one where you're in the time domain EEG and you send a stimulus and you average it many times so that you can see a waveform that's associated with that stimulus and basically averaging out all the extraneous activity that your brain is working on. Yes?
On the Neurosky thing, to your question, we had one of our members, or a couple of our members, build a thing called Mind Flame for Maker Faire Detroit last year, where they, depending on their level of concentration, once they had two people competing, and when one of them reached the level of concentration, it would shoot off a propane flame. So they actually interfaced it through Bluetooth connection into the receiver into an Arduino, and then used the Arduino to fire a solenoid. So it's not as detailed as an EEG, but it gives you, I think, 3 different outputs. One is concentration, one is, and I don't know what the other. It
gives you actually 4 or 5, and in theory, it's supposed to output the raw EEG as well. I just haven't successfully forced their app to do that, which is part of why I want to hack it.
The data sheet seemed to say that, but I asked them, and they said, no, it doesn't do that. So
well, that would explain why I haven't been able to make it work.
Well, it's like, I wonder if there's a different model out there that does,
and
we just didn't have the version that, but yeah, it seems to allude to the fact that you can get that in a bit frame or something, but
sure.
It doesn't come out.
No, it doesn't. So I'm glad I'm not the only one who's had that issue. That's a little reassuring. Anything else? Okay, well, I'll unpack them and let people look at them. We can hook them up. If you want to use your own smartphone, you can download the Backyard Brains app, or you can use mine. Doesn't matter, one way or another. And come up, so I stop standing up here in front of everybody. I would say stop at this point.